this is Brainbow. Basically, the idea is this one piece of DNA will make every single cell that it's in turn a different color. So it's really, really helpful with neurons because you can label each individual neuron a different color, which is fascinating. But the way that this works is frickin' brilliant. And the thing that kills me about this, and the reason that I'm going about what I'll show you, or what I'm hoping will work, and I have no idea if it'll work. So the reason I'm going about this is because this is a ridiculously clever piece of DNA. And in my opinion, if I can make it work the way that I want, it will be the ultimate genetic engineering teaching tool. But somebody just kicked in. Oh, thank you. Thank you, Benjamin. Greatly appreciated. Okay, anyway, but the biggest problem with Brainbow is that it's brains, right? Like, these are neurons. This is a living animal that they had to do this to. And I couldn't, for the life of me, think of an ethical way to do this, not the least as a kid, but even on a video. Like, watching, like, I could modify, like, a fish or something to do this and then, you know, dissect it on camera, but that's not going to go well. Like, YouTube is not going to like that. People are going to get all fucking weird. And I mean, I don't particularly like modifying animals for fun. It's not cool. Like, if you're learning something, great. But otherwise, I don't like it. So I wanted to make a way to do this in a format that the average person interested in biology could actually interact with. Because, you know, like, animal modification is something that really needs to be taken very seriously. One of the reasons I dislike the Odin, because they don't. So if you're going to modify an animal, it needs to be taken seriously. And I thought about, you know, maybe like flightless fruit flies, right? So, you know, you can modify a fly, and then you can make something like this where you can see every neuron in the fly. That's lovely. But again, I mean, working with animals is annoying. Even working with flies is really annoying, and it's really hard. Like, getting the DNA into the animal is very, very, very hard. You essentially have to modify the eggs in the case of the flies, and doing so is super annoying. It's doable, but I mean, you need special equipment, and it's a pain. I designed something else. So I designed this. This was based off of okay, so this one says CMV Brainbow. This one says CAG Autobow. So this is what I based my thing on, but we'll explain why in a second. So the way that this, actually, I want to see Brainbow circuit. No, that's not right. No. Let's just go Brainbow. Oh, no, I gotta look up Brainbow Autobow. Whoop. Nope. Nope, no. Autobow. I think this brought it up. No. Yeah, okay, this is the one that I was looking for. Okay. Okay, so this is Autobow, okay? So the way that this works, these little triangles, I wonder if I can make this bigger. Open image new, maybe, yeah, okay, cool. Okay, so these little triangles are called LOX-P sites. And this first enzyme here is called Cree Recombinase. So basically what happens if Cree and a LOX-P site are in the same, like, it doesn't even need to be in an organism. You can do this in a tube. But if they're if Cree can touch DNA that has a LOX site in it, basically what it does is it will take two LOX sites and schmoo them together. But it kind of does this randomly. So the way this works is this has three different LOX sites, and so the LOX sites have to match, right? So basically, the first thing that happens when this DNA runs, right, is the Cree recombinase is created. Nothing else is created, just the Cree recombinase. So if when that happens, what it'll do is one of three things. Either it'll go P and P and essentially just cuts itself out. So this is the thing, when it schmoos the two pieces of DNA together, it literally cuts out whatever's between. At least in this case, not necessarily. It depends on how you set this up. But so the idea is if P and P go, it just cuts out the Cree and the Cree stops. The way, and so these three things here, the cerulean, Phi-YFP, and MCATE, are three different fluorescent proteins. So at the end of every fluorescent protein is a stop codon. So basically this is what this whole thing does is it essentially just shuffles the DNA around and it randomly picks one of the three colors to be produced. So if you go P to P, you get the blue. If you go 2 to 2, you get the yellow. And if you go N to N, you get the red, right? Hopefully everybody's following along so far. But basically it progressively deletes up to here randomly. But it only does this once. So if this happens, so basically once it cuts it out once, the Cree is destroyed, and then it stops, and then that's just the way the DNA is in that cell forever. Now, yeah, it's kind of like a DNA loot box. That is a good way of thinking about it. So this is CAG Autobow. This is the like doodle that you put in the paper. This is what it actually looks like. Like this has all the extra stuff in it that actually makes it work. In here, so the Benchlink, for whatever reason, didn't like one of the labels. So Cree is actually in this spot. It's just not labeled at all for some reason. Don't know why, just is. But so basically I took this same structure where you have, a general promoter, right? This turns the gene on. Then you've got the three lock sites here, then Cree, and then three colors, right? These are, they're not, the colors don't match what they actually are, but that's fine. So what I did was I took that same basic structure and I redesigned it to work in yeast. Everything has been redesigned to work in yeast. So allow me to zoom in a little, shall we? So clicky click there, do the thingy. Okay. So that's the terminator. Okay, so yeah, so you see the LOX P sites here, LOX P, LOX 2272, and LOX N. So these are the three different LOX sites. Here's the Cree. And you'll see that it says Piscia optimized. So basically, one of the other problems with this is that every single piece of DNA in here was designed to work in humans. Or not humans necessarily, but animals, right? Which, for those keeping track, a yeast is not an animal. That would make beer very, very weird. But, so the idea is I had to basically retune everything to work better in yeast. Because yeast, every organism is picky about the way that the DNA is. Basically, it works exactly the same way that I described previously with one caveat. So you'll see that there's actually 4 colors here. So when it comes to fluorescent proteins, they're not just fluorescent, right? With the exception of maybe like the light blues or light greens, they are colorful in white light as well, right? So like a YFP generally looks yellow. So what I realized was that if I design this correctly, it should look colorful regardless of whether or not you actually have access to a fluorescent microscope. Because one of the biggest problems with Brainbow is that these are all fluorescent, and this is actually a composite image. This is not what this looks like if you just look at it. This is a composite image of 3 different images under a fluorescent microscope using 3 different filters and 3 different light sources. So that's why it looks so nice and clean and pretty, but it doesn't actually look like this at all. So I was thinking about this going, okay, well, how do I make this so that it's accessible to everyone? You use yeast. Okay, but now how do I make it so everybody can see the frickin' result? So like I mentioned, The light blues tend not to have any color in white light. Like they glow a brilliant blue when you put a UV light on them, but beyond that, they look like nothing. So it would just look like the same yeast cells as everything else. This is not particularly helpful. So what I did was, what I think is really clever, basically I put together two different blue proteins. So this one that's called IRA-CFP will actually glow blue when you put a UV light on it, but a MIL-CP doesn't glow. It just is blue. So this way, oh, somebody, well, thank you, Philip, greatly appreciated. So basically, the way this works is this little P2A right in the middle is essentially like a little genetic hack where you get two things for the price of one, right? So when this does its like randomization, you know, spin the wheel, pick a color thing, if it lands on blue, instead of making one protein, it'll make two. One that glows blue and one that is blue. So this way, if you don't have any access to like a fluorescent, like a UV light and a filter or whatever else, you can just look at the thing and it'll be blue. Now, there's one other caveat to this whole thing. So when you look at Autobow, you'll notice that these things look like there's more than 3 colors, right? Like you're like, but if there's 3 colors, how do you get a pink? How do you get an orange? The answer to that is what happens if there's more than one copy of this DNA in the cell. So if there's more than one copy, what can happen is this randomization process can happen more than once. So if you've got 2 copies, it'll pick 2. And you know, if you get a blue and a blue, it's really blue. But if you get like a blue and a yellow, now it looks green. Or if you get a yellow and a red, it looks orange. Or a blue and a red, it looks purple. Right? So that's the trick to how this gets like, you know, dozens of colors for the price of 3, essentially. It's really clever. I freaking love this system. It is so, so clever. So as a teaching tool, the amount of stuff that I just covered is insanity. Like it's, you know, all the, like multiple different markers, Cree recombination. But the other things that would be really, really neat, because this is a yeast DNA, this is yeast optimized, specifically Piscia, which is one of my favorite yeast to work with. Do you see how this says AOX1 promoter? This promoter is sensitive to methanol. So Piscia generally eat methanol, so of course they need something to tell them when methanol is present. This gene hijacks that system, and so this whole construct will only actually run when the yeast is given methanol. So the trick to this is what you would do is you would take this piece of DNA once it's made, you put it into the yeast. You can then select the yeast that carry the gene using antibiotics. Or, I mean, you could use antibiotics. This also has histidine synthesis. So I could actually sell the kit with no antibiotics. It just means I need to get a lot of the media, which is not so bad if you're buying in bulk. So the idea is, you know, you plate the yeast and select the ones that carry the gene, but it doesn't run yet. Then once you've got lots of cells that carry the gene, you can streak them out onto a petri dish, and the colonies that form, if you streak them out onto media that contains methanol, all of a sudden the gene turns on, and then every colony should be a different color. So it's essentially like rainbow yeast instantly, all on one plate from one piece of DNA. And the reason this is really cool is because the way that this particular plasmid works is it's an integrating plasmid, and more than one copy can integrate. So what this means is that not only would I get to teach Cree recombination, inducible and constitutive promoters, multiple reporters, I could also teach transformation and integrative transformation. This is crazy for the amount of genetic engineering that you can cram into one thing. So I think this is going to be amazing. Now, the cherry on the top. Are you ready for this? Every single one of the fluorescent proteins is not patented. So this is super duper duper cool for a lot of reasons. What I didn't realize was most fluorescent proteins are still under patent. So a lot of them are expiring soon, but a lot of them aren't. And there are certain companies that will sell you various plasmids that contain the fluorescent proteins so that you can use them for whatever it is you want to use them for. But the problem with this is they will sue the shit out of you if you do anything that's outside of their terms of service, which I hate that. So I specifically chose fluorescent proteins that are patent-free. Why is this awesome? The reason this is awesome is because while I was designing this, and I kid you not, designing this took literally all frickin' day. So this is why I'm not doing this one live. I don't have the time. It took me a whole day. So the best part about this is when I designed this, do you see how this is highlighted, this up high here? This is a restriction site. This is a convenient location where if you add a specific enzyme to a pure sample of this DNA, it will cut right there. Like right on this, like, do you see the little wiggly line? That is exactly where the enzyme cuts. And if I scroll down, you'll see that there's another one right here. And there's another one over here. I put a restriction site on the ends, a different restriction site, on the end of every single one of these proteins. This is very cool. Well, thank you, Christopher. Greatly appreciated. Yeah, basically, I put restriction sites on the ends of every single one of these proteins. And the reason that I did that was so that I could make this, which is basically it's essentially just cutting out the fluorescent protein and sticking it into the same backbone plasmid, but that's empty. So it doesn't have any of the rainbow stuff, but it does have the plasmid with convenient restriction sites on both ends. So there's a convenient restriction site, there's a convenient restriction site, and these restriction enzymes are dirt cheap. So I want to carry on the site an entire line of fluorescent proteins optimized for yeast. So if you ever wanted to say, once you're done with the kit, you're like, yes, I understand yeast genetic engineering now. I would like to do some of my own. You can buy the individual colors, or you can just buy them because they're cool. But if you wanted, you could buy them, cut out the fluorescent protein, and then stick it into your own plasmid of your own design and do whatever you want with it. I don't care. That's what it's for. Eventually, I'd like to have a line of the same thing in E. coli, but a friend of mine is kind of doing that already, so I don't want to compete. So if you want this same service, because we're using all the same proteins, because we want to, specifically because we're big on open source and sharing and all this kind of shit. So if you want a yeast optimized version, you can come to me. If you want a bacteria optimized version, you might be able to go to him. I might set something up so we may do a collab store thing. I don't know yet. We're got to work on that. But anyway, the point is you're going to be able to buy colorful yeast plasmids. Now, the caveat is these do not work in food yeast. Piscia is not food. I specifically did that because I don't want people eating this. While the glowing beer is very, very cool, and rainbow beer might be nice, if I do that, then there could be like legal issues of selling a thing meant for human consumption that's genetically engineered, and that's no bueno in Canada. So I put it in a yeast backbone and yeast that is not food. However, it is yeast optimized. So if you wanted to take it out, put it in a yeast backbone, which I might carry, and do your own thing, that's on you. I didn't sell you this. I just sold you the pieces. If you choose to do that, that's on you. So yeah, that's this. Okay, a couple of questions because I'm seeing them kind of go by. What the hell? Okay. The double blue won't. I don't think it'll mess up the color combinations, at least in white light. But it doesn't glow in white light. So under UV, like if you're trying to do like a fluorescent color combo thing, it still wouldn't matter because it doesn't glow. It's just protein for all intents and purposes. Somebody calling me? Should no. So yeah, that's this. Once all of these are done, these will be, and done, I mean synthesized. Like once the company has made the thing and sent me the thing, and I test it out and I make sure it all works, first I'm going to do a video about it. But then we're going to mass produce it. I say mass produce. We'll start with like 100 and we'll go from there. We're probably going to do basic, kind of like merch drops, where there'll be limited supply, and then once it's gone, and it'll take us time to make more. But this way, we don't need to scale up to insane levels and then not sell anything, so we can kind of start smaller and then work our way up. So that's these ones. There are a few more, and I actually have my list, so give me a second to just pull it up, that I wanted to talk about. The egg and the milk plasmids. So these were one of the first ones that I designed on stream, and here I'll pull them up so I can show you. Milk and eggs. Here we go. Is this the right one? No, this is not the right one. It's the other one. Okay, so anyway, milk and eggs. So for this, I created two plasmids. This is the first one. Zoom that in because that's a mess. So do you see how this says ovalbumin? Ovalbumin is the main component of egg white. And the idea with this was that you could grow egg whites using yeast. So I tested it. It kind of works, but there was, I think what the issue with this was I was a little, what's the best way to put this? I thought it would work better than it did. So I was expecting to get way more ovalbumin production than actually happened. And this, I mean, this could have been a lot of things. This could have been the strain that I used. This could have been the media that I used. There's a lot more like optimization that I would need to do. But the point is, the media did not turn into egg whites, which is what I was hoping. So that was a bust. So in order to actually use this properly, the best way to do that would be, I need to stick a tag on here so it's easy to extract it and purify it, which was not what I did originally. So the egg plasmid will probably be up for sale at some point, but one of the things that I'm going to do today is I'm going to fix this. So what would have been smart, which I did not do, but what would have been smart, is if I added a special tag on the end of this protein that will stick to a specific basically like a metal complex. Like you can essentially buy it's like a tube. It's a column with little beads in it, and the beads have a special metal in them that this tag will stick to temporarily. So like if you run your media through this, only this protein will stick to the beads, and then you can wash all the rest of the crap out. And then once that's done, you run another solution through it that makes all the protein let go and come right out. So that way you end up with a super duper pure protein with none of the extra junk, and then you just kind of like redissolve it in water and you're good to go. Yeah, people correctly pointed out this is called a his tag. I don't know if I'm going to use a his tag. I might use a different tag. We'll see. But yeah, so hopefully, depending on how long this stream goes, because I mean I am getting hungry, so I don't know how long I'll go for. But one of the things that I need to do is I need to add a his tag to this. But before we do that, I want to show you something a little different that we haven't covered on streams before, and that's how I actually order my DNA. So I use a company called Gene Universal. They're fantastic. They have some specifics of how you need to interact with them, but I'll show you. So the couple of things. First, their service is a little slow unless you pay for like the fast service, which they do have a fast service. But these guys can make anything. Like I've given them like highly repetitive natural proteins, unfucked with, like straight out of the organism, like here you go. And they printed it just fine, which is insane because if you tried to do that on a company like Twist, Twist just kind of shits themselves and goes, I don't know what to do with this. So these guys are fantastic. However, like I say, there are some caveats with working with them, which is I don't actually know where they're based. I think it might be China. I'm not sure. But the point is, you basically need to tell them exactly what you want to do. They don't have like an online tool like Twist where you just like submit a thing, because it's an actual person who receives the request, analyzes it, and decides whether or not they can make the thing. So this is the way that I have always written my orders, and it's only ever been a problem once where there was like some weird shit in the plasmid, and we'll get to that today. Actually, maybe today, we'll see. Okay, so when I want them to make something, I go over to Benchlink, and for those of you who are asking, this program is called Benchlink. It's an online tool. They do have a downloadable one, but it's like 10 grand, so we go with the free online one. I don't have a basic attention tipping code, so thank you, but I'll have to set that up. Anyways, so let's go to Auto Yeastbo. Okay, so I want them to make this chunk, right? So basically from this promoter, I want them to make from about there. Oh, I clicked on something by accident, my bad. So I essentially want them to make from that all the way to the end, which this, oh, come on, fuck. So you scroll all the way to the bottom. That's the terminator, that's that. Okay, now that's part of the thing. Basically here. So, what happened? Okay, fucking. Okay, so I basically want them to make this whole giant chunk here. So before I actually ever put a piece like this into a backbone, here I can show you. Just got to go to Brainbow. Okay, so you go to Brainbow, insert, Yeastbo core. So when I'm designing DNA, I don't usually design it in the backbone. I'll keep in mind what restriction sites I need on either end, but I'll design it as a blank file like this and just build it up piece by piece. So in this case, I want them to essentially print this entire file. So that way, if they take this printed chunk and digest it with the right enzymes, they can shove it into the backbone that I want. So when I'm writing an order, I literally go like Command A, Command A, just copy, copy. Okay, then I go over to literally a Word document, which I realize this sounds insane when you're talking about like super high-tech DNA printing, but yeah, a Word document gets it done. And in big, bold letters, I say, print this sequence. Literally like, print me this. And it's just unformatted, there's just random, capital, lowercase, doesn't matter. It just gets parsed. So it's just a big wall of text of, make me this. So it's like, okay, great. They've printed that. What's the next step? I tell them, okay, I want you to take this piece and I want you to digest it with these two enzymes, right? So BAMHI and NADI, which if we go over to Benchlink, you can see BAMHI is on the front end here. And if we scroll to the bottom, NADI is right here. Perfect. Where would I have that? I don't know if I have a blank. Oh, I know where I would have a blank. Colorful yeast. Okay, this is the backbone. This is what it looks like before you put anything into it. So there's this little thing that says MCS. Might be a little hard to see, but this is called the multiple cloning site. So it's literally a little spot where they've put multiple restriction sites. Specifically so that it's very easy to pop the thing open, shove in the thing that you want, and call it a day. So what I did was I said, okay, this is lovely and all, but I don't actually, so one of the perks of this particular plasmid is it has this chunk in it, which you'll see as alpha factor secretion signal. Basically, this tells whatever protein is stuck to this, get out of the cell. So basically it makes the cell poop it out, essentially. This is really, really helpful if you want your product to not be inside of the yeast at the end of all of this, but in this case, we don't want that. So instead of using the multiple cloning site or one of the enzymes that's in here, instead I scrolled up past that, grabbed this restriction site, and said, I want to use this one. So I'm going to use BAMHI and naughty, which means it's going to cut all of this out. It's gone. We don't need it. So now if I go back to my Word document, like you can see, it says digest with BAMHI and naughty. Those are the two spots where I would like them to pop the DNA open. Now I tell them digest this backbone. That's the one I just showed you. It's called PPIC9K. And these are the enzymes that I want them to use to do that. Cool. So now what we have is we have a backbone which has been digested and it now has dangly ends ready to accept an insert. And we've got the insert printed with dangly ends ready to be connected to the backbone. And I say ligate the two pieces together. This means take the thing you printed, take the backbone, now put them together. Once that's done, technically, you should have a finished thing. But what I always put to make sure that they know this is exactly what this is supposed to be is I'll say the result should be, and then I'll highlight the printed sequence. So like I'll copy this and I'll literally highlight it in the backbone and I'll copy the entire thing. So if I can find a auto USPO. So I'll literally go on this file, I'll go like Command A, copy it all, paste that in, and then go back in and highlight the thing that I had them print. So this way they know exactly where the thing I told them to print should be and how it should look relative to the rest of the backbone. Because as they're making this, they'll actually sequence it. And that's part of what you're paying for when you have them make something like this. They'll sequence the whole thing to make sure that everything is perfect and all the letters that are supposed to be are there. Because with DNA, if there's a single letter wrong, you're fucked. It just doesn't work. It can break everything or so much worse. So basically they're doing a lot of checks. And so basically I'm giving them all of the info that they're going to need to do those checks. So basically what this has now said is print the piece, take the backbone, digest both, put them together. This should be the result. Now they have essentially, like if you're in my head, I'm thinking through all of the steps that they're going to go through to actually make this. And so in my head, I'm like, okay, they're going to have literally a tube of clear liquid with a tiny amount of the perfect DNA that I told them to make in it. That's not really helpful. I kind of need a lot of the stuff to work with. So what I say is transform the result into E. coli and select with ampicillin. So the reason I say this is because if you look at the gene, or if you look at the plasmid, right, you'll see this bit that says AMP-R. AMP-R is ampicillin resistance. Ampicillin is an antibiotic in the penicillin family. And it's essentially the one that everybody uses when working with E. coli to just select the ones that have been modified. So it has this. It also has an ORI, which is another piece that you need for this to work in E. coli. So basically what this is saying is this plasmid, if you put it into E. coli on plates that contain ampicillin, the only colonies you'll get on that plate contain this DNA. Cool. So now that now we're going to assume that they've done that correctly and they have the DNA in E. coli. Now I say select a colony containing the above sequence. So they're going to pick a bunch of colonies off of that plate and they're going to streak them out, grow up a lot of them, and then extract all the DNA from them. Well, they'll do a plasmid extraction, get the DNA out, and then they'll check the sequence again and make sure that the thing that I had them design is in the E. coli. Once they know that that's correct and it's all working properly, they then take that little sample of E. coli and they drop it into like a 5-liter bucket and they grow an absolute shitload. Once they have that ridiculous amount of E. coli, they run it through a process that extracts all of the DNA out of that massive amount of E. coli. And then I've told them, okay, now prepare and ship 10 times 100 micrograms of DNA. So what this means is they're going to basically they're going to extract enough DNA that they can make a solution that's 1 microgram per microliter. Okay? So essentially what this means is they're going to make, so if they were to put all of this into one tube, that would be 1 milliliter of liquid that contains 1 microgram per microliter in the tube. But when I say 10 times 100, this means they're going to send me 10 tubes, each with 100 microliters, containing a total of 100 micrograms per tube. The reason I have them make me 10 tubes is so that A, I can send off some of the tubes to different people so that if anything happens to my sample, it's backed up. And also, when you're working with it, sometimes you like bump the side of the tube with like the edge of the tip or the edge of the pipette that isn't sterile. And so sometimes like destructive enzymes just from your skin or from the air or whatever can get in the tube. So sometimes the tube might just be fucked and there's nothing you can do about it. And so keeping it as 10 tubes makes it so that if one of them goes down, I've got 9 backups that I can work with. And of course, you know, and so then the other thing I say is, and 1 stab. So basically the stab means that they took one of the colonies that contains the right sequence of the E. coli, and they make a tube full of agar, and they like dip it in the E. coli, they dip like a spike in the E. coli, and they like stab it in the agar. This is just a way of transporting E. coli. So essentially what I'm asking for is I want 10 tubes of DNA and 1 tube with E. coli in it so that I can make more of the DNA myself if I need to. All ready to go. So with that, this file is essentially ready to be sent off to the company. And you send them an email and be like, hi, I'd like you to make this. You make an account first, you contact the rep or whatever. But this is now ready to be sent off to them. I've also started including, how they should label the tubes. Because if you don't tell them label it this, they'll just label it with a number. And when you're ordering lots of DNA, you end up with a freezer full of tubes that are just numbers. And you don't know what anything is. I had the updated version of the lactase thing in my freezer. I didn't know which one it was. I had to talk with my friend who ordered it for me. I'm like, hey, is this the one? I don't know. I've got 100 tubes that are unlabeled. It is an absolute shitload an SI unit. You bet your ass it is. Is the webcam new? Yes, it is. This is a new webcam. I'm doing it on my fancy computer now. I'm in a new apartment. Everything's new. Okie dokie. So this is a basic file. Lovely. But yeah, this is basically it. And so now there was a question about what does this cost? So let's do a little checky check here. Perks of doing this in Word. So this is huge. I'm not gonna sugarcoat this. This file is massive. So there are about 5,000 characters, give or take, which means approximately 5,000 letters. So if you go onto Gene Universal and you go on to Services and Custom Gene Service, they've got a 20% discount going. So, if you need some DNA, now's the time. So you scroll down and you look for this. So it tells you what it costs. So if you want to print something small, it's real cheap, real cheap. It's $100. If it's less than 500 base pairs. So if you want an itty bitty protein, $100. If you want bigger than that, they charge you 19 cents per base pair. Now, like I say, this is, compared to Twist, so Twist Bioscience is the company I was referencing before. These guys charge something like 9 cents per base pair. So they are far cheaper and they're very fast, but they are extremely picky about what they will and will not make, which is super fucking annoying. Whereas these guys, they charge a little more, but their quality is incredible, and I've never had an issue with them. So, you know, above 5,000 base pair, they'll just give you a quote, but we're going to use 25 cents just as a ballpark there. So we're going to call it 5,000 times 0.25. It's about $1,200. But, you know, it's 20% off. So it's around 900 bucks, give or take, American. So, I mean, I'm not gonna sugarcoat it. This is expensive. So I've said this in videos before, and I'll say it again, and I'll say it 1,000 times more. Biology is very fun. It's very cool. It's lovely that more and more people want to get into it, and I highly encourage this. However, biology is not a poor man's sport. Everything is expensive. There is no cheap solution to anything. Everything is expensive, and if it's not expensive, it's going to cost you time and lots of it. So I've taken to sticking to, I'll pay a little more if it actually gets done and it works. Because if, you know, you might take the cheaper solution, like maybe you go with Twist, but with Twist, they'll only print your gene. They won't put it into a backbone for you, and if they will, it's only their backbones. Otherwise, there's like a $600 fee to submit your own backbone. Whereas with GU, you'll take anything. Like, you want to submit a new backbone? Fine, send it along with your order. You want to, you know, have, and Gene Universal saves every sample that they make for at least a year, if not 2. So if you want to like update a file, or you want to like work with something that you've already made. So say I wanted to work with the yeast bow thing that I've made here. I could just send them an email and be like, hi, start with this, and I can just give my order number, and they'll just pull it from their stock. I don't need to send them a fresh sample, which is really, really helpful. So, yeah, but anyway, so this is expensive. This is a very big gene. I don't normally make stuff that's this large, but because I see this as such an amazing teaching tool, I think it's worth it. So yeah, it's going to cost me a lot, but I mean, this is why we have the support of you lovely people who occasionally donate, and ad revenue and all this kind of stuff. But it is expensive. It's why it takes me a long time to order these sorts of things, because it's a lot of, I've got to plan it out, think it through, quintuple check everything, because if there's a problem, it's very expensive to fix. So you got to make sure it's right the first time. And I have made many mistakes in genes that I've had synthesized, and it has cost me a shitload of money to deal with it. And it really, really sucks when that happens. So, you know, there's 2 ways to learn a lesson, the easy way and the hard way. And I've almost always learned it the hard way. Yeah. But anyway, so that's this. This particular one is going to cost a lot, probably around a ballpark of $900, but the other thing with GU is if you work with them a lot, they'll just give you discounts occasionally. Like if you're a customer that's returning and you're constantly ordering stuff, like they'll give you discounts if they fuck something up, or like your design doesn't work. Because I've only had this happen once, where I asked them to make something and they just couldn't do it. It was too weird, or just something didn't print right, or whatever. Rather than just sending you garbage and charging you full price, they'll usually either give you like 50% off, or they'll give you credit towards your next order, which I really, really appreciate. And again, none of this is sponsored. They're not paying me shit. I'm paying them lots of money. I just really like them as a company. Like they do good work, and I appreciate that. So somebody's asking, Gideon, what is this sequence? Or yeah, Gideon asked, so what does this sequence do? You're going to have to rewind. I'm not explaining it again. It's really complicated. What else we got? Okay, cool. So with that said, and this covered, that's this. Now, like I said, I put restriction sites on the edges of every single one of these proteins. So I've already done most of the work for this, because this took me like an hour. So basically what I want them to do is I want them to take this printed thing, and I essentially want them to cut it to ribbons. And I want them to isolate each of the individual fluorescent proteins or chromoproteins, and then stick them into their own backbones. So let me show you what the synth file looks like. And this isn't finished, and we're going to finish it on stream, just so you can, see what's up. So basically, somebody just donated. I seeing that number correctly? Did somebody just donate over 1,000? What the fuck? Thank you. Thank you, Pendleton Ward. Wait, Pendleton Ward, you're not like the Adventure Time guy, are you? Because that would be just the wildest. Oh my god. Thank you. Thank you so much. That's my brain ground to a halt. Oh my god. Thank you, dude. Yeah, there's no sound because I stopped talking. Holy crap. Thank you. Pendleton, you the boss. But seriously, if you're the Adventure Time guy, please let me know, because that would be really cool, and I'd be a huge fan. I'm just trying to keep my eye on the chat in case he says something, because I want to know if that's the real Pendleton Ward. Because over the years I've gotten some people to subscribe that it just makes me chuckle every time I see them. I'm just like, what? Why? Like what? What's his name? Hamilton something, the drug guy, the guy who does all the drugs, and did the Vice show. He's a subscriber for some reason. I think Lockpicking Lawyer is, and I'm just like, what the fuck? It's great, but it's always surprising. So again, thank you, Pendleton. That was awesome. Shit. Okay. Fuck. I guess I should get this done then. Okay. My print cost is now covered. True. Okay. Let's get back to it. Thank you, Pendleton, again. Yeah. Wait, did you reply? Pendleton Ward. For science. Cool. All right. He didn't say if he's the Pendleton though. I don't know if he's the real Pendleton. But seriously though, thank you so much. Also, Dark Order of Magi. Thank you very much. Greatly appreciated. Thank you everybody who's donated. It really does. I really do appreciate it. Especially when I'm not making videos. I'm just hemorrhaging money building the new lab. So this is really, really appreciated. So yeah, this is great. Oh my god. Okay, I'm still floored. So if he replies, somebody let me know. I want to get back to the stream, but I also want to know if he's the Adventure Time guy, because I'm such a huge fan of that show. Okay, back to work. Doing the thing, not being distracted by my awesome fans. Okay. Right. Okay, so what I was saying. Basically what I want them to do is I want them to take this plasmid and cut it 4 times. So not all at once, you wouldn't do this all in the same tube. I would want them to do this separately because it'd be impossible to separate out the pieces. But in order to get these pieces to fit into the backbone, which is this, as a reminder. So I need every single one of those to fit in this hole. The problem with that is I had to use different restriction sites. Definitely need to give Pendleton a sample. I mean, send me an email. I'll sort that out. But anyway, so I need to get all 4 different proteins to fit into this hole, but like trying to get a square peg in a round hole, they don't all fit. because they don't all have the same restriction sites on both ends, which is obviously a problem. One of them does. Like one of them, the red one, where's the yeast bow core? So E for red, this one has the correct restriction sites on the front and back, allowing me to immediately slot it into where it's got to go. And I did this for a specific reason. That reason is a friend of mine challenged me to a basically like a race, essentially. So it's the nerdiest race ever. Basically, we each have organisms that we prefer working with. So he prefers working with E. coli, I prefer working with yeast. And basically, the race is in 1 liter of media, who can make the most protein. And for the sake of ease of quantification, we used E for red, which is this vibrant pink. I'm going to show you E for red. Hopefully it just comes up. Images, yay, okay. It's this ridiculously pink protein. So the idea is we can basically just take a sample from the media and quantify how pink it is, like look at it on a spectrometer, to basically figure out how much protein is in there. It's for convenience, and it's a small protein, so it's easy to make lots of. Jakub says, please tell my friend Steven to go to bed. He has an important meeting yesterday. Yesterday? He has an important meeting yesterday. What? Well, Steven, go to bed. You've been told. Okay, so anyways, back to the thing. So as part of the race, we're in 1 liter. We've got to see how much protein we can make. And we have to make the identical protein. So like amino for amino, they have to be the same. But other than that, there's no real rules. So basically, I made sure that this just slots right in to the backbone, so that way it's the easiest. Just cut it out, stick it into the new thing, we're done. So for all of the other ones, though, you run into a bit of a problem. So because it's just not going to fit. It will not, right? Like the pieces don't stick, and if they don't stick, it's no good. So I have to make adapters to go from this restriction site to whatever's actually on the protein, right? So like if I go to the yeast bow core, and let's pick the Blaze, for example, Blaze YFP. So this one has an SPEI site, but I need the front end to be EcoRI. So obviously these two things they don't schmo. So to fix that, I essentially had to make adapters. So this is the adapter here. It's this piece here. Basically what I'm having them print is essentially this one from here. Come on, all the way. So basically, I need them to print this little piece, and then you can, like, once they isolate the big chunky, they print the adapters, they cut the adapters, and then they can stick the adapters onto the piece that they isolated, so that way the whole thing fits into the backbone. Hopefully that makes sense. So because the red one just fits in directly, that means I only need to make 6 adapters. So if we look at the synth file, here are the 6 adapters. Of course, I'm a turnip, so I didn't actually label these when I put them in. I know they're paired up, but I don't know like which is which. So I'm gonna have to check that. But okay, so let's go through this. So the things that they have to print are just the adapters, right? So 6 little teeny tiny pieces. Hopefully this doesn't count as 5 separate genes. Hopefully they just, cut me a break and just say this is one thing. But then what I'm telling them to do is take this. This was the order that we just went through, which, remember, I told them to call it Auto Yeast Bow V1. So I said take 4 samples of Auto Yeast Bow V1, right? So now they're going to have 4 tubes. Each is going to have a little bit of this DNA in it. Then I tell them to digest each sample with 2 different restriction sites, which is going to cut out individually the 4 different proteins, right? So you've got and I color coded them to make sure that I can keep track. And because there's a lot going on in this one file, I need to make sure that they don't get confused. Because the last thing you want is to like be like, I'm transforming a red. It comes out blue and now you're confused, or something doesn't work and you want to know why. So it's important. So basically cut out these 4 pieces. Great. Then I say digest 4 samples of the backbone, which is the same backbone we used before, using EcoRI and AVR2. This is the reason that they only have to use the same restriction sites each time is because of those adapters. The adapters are made to make sure that, you know, everything goes the way it's supposed to. Now this is the bit that I need to fill in. There's 2 bits that I need to fill in. Somebody else donate? Oh, thank you, Bubblink. Bubble G? Bubble G. Thank you. Greatly appreciated. I'm glad you like the streams. So, you know, there's a couple of things left in here that I gotta sort out. I've already written the, you know, the ending bit of, you know, transform each, select a colony, prepare 10 by 100, and then how to label everything. But the last thing I need to fill out is what the results should be. So I'll show you all the plasmids that I've designed. And I need to fill out this of what fragment needs to be printed with what, or needs to be digested with what enzymes. Just quickly, the Better Game asks, what's the best way to donate to you? The best way to donate is via the link in the description, the Streamlabs link. That goes basically directly to me. If you donate through YouTube, they take like a 30% cut because they're bastards. So for no reason. So yeah, if you'd like to donate, it's greatly appreciated, and the link in the description is the best way to do that. Thank you very much. Okay. Just anything else? Quentin, no, we've already done that. This is something else. This makes rainbow yeast. Would one do this if one made, okay. Benjamin asked, would one do this if one majored in microbiology? It would very much depend what kind of microbiology, but almost certainly no. The thing that you want, if you're looking for like a degree that will teach you this, you either want something that says synthetic biology, molecular biology, or maybe biochemistry. Microbiology, you might do some of this, but probably not. It's more like studying microbes as they are, not really messing with them too much. Yeah, so that's your answer, I think. Okay, now back to the thing. I need to find which of these adapters need to be digested with which things. So let's do that. This one, ACT-AGT, I think is the ACT-AGT. Yep, it's this one. Okay, so I just happen to remember that because I was looking at it a second ago, but this one, which I think was number 3, needs to be digested with SPEI. So that was number 3, so we go down to here, SPEI. Lovely. Now we scroll down. We're looking for CTT-AAG, which needs to be digested with AFL2. I don't know if I'm going to remember that, but we're going to give it a go. It's probably the one right next to it. Did I? CTT-AAG? That looks right. And it needs to be digested with AFL2. So we go down here, we go AFLII. Whenever it's ambiguous like this, because L's and I, like, restriction sites are named poorly, to say the least. So whenever it's ambiguous like this, to make sure they don't use the wrong one, I'll use like capitals or something just to really make it clear. We'll talk more about the milk and eggs later. Somebody just donated. Thank you, KR. Thank you very much. Greatly appreciate it. Okay, moving on. So we now have SPI, AFL2, which means 3 and 4 should be fragment C, which should be yellow. That's not good. Huh. This is why we check. Okay, so this is the problem when I'm trying to get them to do something complicated like this. It's a frickin' mess, and just keeping track of everything is annoying. So I really should have taken more care with that. Let me just double check something. Yeah, CTT, AAG, yep, that's correct. AFL2, yep, that's correct. Okay, so these two, so that I don't forget, I'm going to highlight in a lovely yellow. Fuck. Okay, that means I need to scroll down. So, okay, so fragment B is my yellow. So that means I need to scroll down here. Isolate, yeah, ligate 1 and 2, isolated fragment B. No, that is not correct. This should be 3 and 4. I don't know why I pasted them in like that, but I did, and so we're fucking living with it. Now we need to figure out what these other ones are. So let's go back through. Let's pull up my MLCP. Where's the X by end? It's here, I think. Yeah. That should be all of it. Yeah, okay. TCT AGA. We're looking for a TCT AGA. On which end? I don't see it. Oh, TCT AGA. It's right here. Okay, so that means these two are going to be my MLCP, which MLCP is blue, I think. Yeah, okay, well, so this one's supposed to be XBY, so we'll do that one first. So number 1 is XBY. Somebody asked, what was the biggest oh shit moment you've had during one of your projects? Okay, I'll tell this story really quick and then we'll get back into this. I was working on a really crappy DIY vacuum chamber that was made out of a mason jar. And I was doing some plasma stuff in it, and I connected a wire to the wrong part, like to a part that should not have gotten hot. And it did get hot once I turned the power on. And I distinctly remember hearing the most horrifying like clicking noise of, I could hear the glass breaking. And I knew exactly what was about to happen, which was the glass was about to implode. So literally, like, before I literally had enough time to think, oh shit, and then dive out of the way before the whole thing went thunk, and like glass went everywhere, and like the top sucked into the bottom and dented the base. That was a nightmare. That was the biggest like true like oh shit moment that I've ever had with a project. But I dealt with okay. The only other like oh shit moments weren't really like that. Like they happened, and then I realized I was in trouble, right? Like there wasn't, like I didn't have time to think about it. Before I knew anything about chemistry, I was being really dumb and like heating sulfur in a sealed container until eventually it just popped and like sprayed molten sulfur at my face. Not fun. Don't do that. Very stupid. But that was what happened. All right, scrolling down, this should be FSci, GGC, CGGCC. Okay, GGCC, GGCC. Yep, that's perfect. Okay, so that should be, I said FSci. FSci, lovely. Okay, moving along. FSci. Lovely. Okay, that should be all of it. But so if C is XBY and FSci, that means C needs 1 and 2, which is good because those were the only backwards one. 1 and 2, which means this should need 5 and 6, which means we are good. Okay. Okay, give me a second. Somebody just donated, so thank you, Amaritha. Hopefully I'm pronouncing that correctly. I just want to see this. Says, I dropped out of bioinformatics and genome research to become a translator, and I never regretted it. Still, it makes me so happy to indulge in my passion through watching what you do. Thank you so much for your content. Well, I'm glad you enjoy it. Thank you very much. Greatly, greatly appreciated. Okay, cool. So we've almost got this sorted. I've got 4 out of 6 ready to go. which should mean if this is a mil CP, which is blue, which is why I changed the color to a darker blue, this one should be the CFP. So I should just be able to go to here, grab this one. Yep, there we go. So we're looking for GGGCCC, which should be a pi. GGGCCC. Interesting. Why is that number 6? I don't know, but here we are. So number 6 needs to be a pi. Okay, that's backwards as hell, but whatever, working with it. So number 5 should be the other side, which is over here. Right. TACGTA. TACGTA. I forgot about this. Okay, so this one is snobby. Okay, do that. Go here. Snobby. B. Lovely. Okay. Finally got that done. That was super annoying, but it's done. We're done. Lovely. Okay, so we now have, I'm going to highlight these so I don't forget, because I am a forgetful man. So that one's got to be that color, which means this one needs to be, excuse me, got to sneeze. Okay, cool. I know. I like doing everything color-coded because it just makes it so much easier to keep track of everything. Because like it's so easy to fuck something up when you're doing this. Okay. Cool. So this is where things get very, very messy because I've got, this is why I saved this step for the very end, where it's, all the results should be. It's just pasting in huge chunks of code. So it, I always save this for the end because as soon as you do this, it makes scrolling through this a nightmare, especially on a file like this, where I'm essentially asking them to make me 4 different plasmids in one file. I've never asked them to do something this complicated before. So I don't know if they're even going to accept it, but if I write everything out really neatly, it should be fine. What do we got? Cool. So somebody asked, is he expressing all colors under one promoter? Technically, yes, but also no. The thing I'm designing right now is 4 separate plasmids with 4 separate colors. But the thing I made before randomly chooses one of 3 colors and then expresses it. Okay, so also with the rainbow yeast thing, I don't know if the individual colonies will look a specific color. I hope that they do. If it works the way that I think it will, that should be what happens, but I'm not sure. But the individual cells will be all individual colors. So if you take a little sample of any of it, stick it on a microscope slide and look at it, it should all be rainbow colors, which should be very neat looking. Okay, lovely. So now I need to do all the results should be things. So in this one, I'm saying ligate isolated fragment A with digested PPIC. This is that first one that I mentioned, the pink one that just slots right in. So now if I go to finished plasmids, assuming I have actually done the thing, I can grab this. I can do this, go Control A, copy, and now I can just paste that in there. Why is that bold? I don't know. We're going to unbold this. Bold. Now I'm going to grab this. I might need to trim some letters off the end. This is going to take a sec. If I do that, nope, still doesn't like it. Why do you not like this? Why is it not finding this? Well, that's weird. Because it should be this. Right? It's the only bolded thing. Why do you not like this? That's very weird. I did copy it off of the right thing, right? Yeah. So what the hell? Why you no work? Why you no work? new plan, copy. Okay. Well, then what the hell is this then? Why isn't it showing up in AATT? The fuck is this? What? Okay, well, and this is why we go back and check everything. What the hell? This is very confusing. I don't know what went wrong. Okay. Okay. I see all of this stuff. ATG. ATG TCA, right? ATG TCA. That's correct. This is correct. Which means there's a bunch of extra schmo. Oh god, no, go back, undo, undo. Very undo. What? Because, like, by all counts, this should be the thing. Right here. I'm going to just copy a smaller section. Why does it only find one? That doesn't make any sense. That makes literally no... Okay, I'm going to just copy a section from the middle. Copy FV. Now it finds two. Where's the other one? Okay, so it is right. Sorry, this is very frustrating. I have no idea why it just did that. That was more than a little concerning. What the hell? Maybe I copied wrong? AATC. What did I copy incorrectly? G. TTC. Oh, I didn't copy the G. But still, that shouldn't affect anything. Wobble up a dub dub! Nice. Thank you, Knucklehead. Greatly appreciated. Mosey asks, why are you obsessed with expressing different fluorophores? I'm not. The reason that I'm doing this is not for my own use. I mean, having these colors is lovely. I you know, I like rainbow stuff. And, you know, rainbow biology is very cool. But the reason I'm making this specifically is, like, so this thing, the construct that you see right now, is literally the best genetic engineering teaching tool I could think of. And making plasmids that carry the individual proteins is not useful to me per se, but it is well, I mean, it kind of is, because I can use it for my own projects if I so choose. But it's more useful for other people who might want to work with fluorophores in yeast. Did I copy a space before it? Maybe. I don't think so, but maybe. Copy FV. What the hell? What's on the back end? Did I copy a bunch of extra schmo? Maybe. Because this is in here. It's not supposed to be. Copy. Sorry, this is just going to take me a second to sort. See, that's concerning. The fact that that chunk shows up in here, but not in the bottom, is concerning. Why is there extra stuff in there? Because if I did this right, it's supposed to go from there to there. Copy. That looks real different, doesn't it? That looks really, really different. But now, if I go copy FV, it's still the only one in there. What the fuck? What? Come on, man. Okay, this might... I mean, maybe this doesn't like the restriction sites. Let's cleave some of that off. Cleave some of that off. Right? Like, at which point does it freak out? ATTC. Right? Right? I'm copying the right thing, so why the fuck isn't it? Outstanding. Okay. Okay, let's go to, I had this, FV, 0. Why is there 0? Was there always an HEI site in there? Because I don't remember there being one. Did I change something? Okay, I feel like this is probably getting boring for some of y'all. So I think what I'm gonna end up doing is I'm gonna have to futz with this off screen because I don't know what I did. I think probably late night, I decided to like shove some extra stuff in here. Like, you know, adding an extra restriction site or something just to make it convenient, because like this restriction site is not here. So I must have had to add something to make it work with the backbone plasmid that I'm just forgetting that I did. So we're gonna have to cycle back to this at another point, because I don't want to, I mean, we've already been going for almost 2 hours, so I'm gonna have to wrap the stream up soonish. It's not a matter of what I copy pasted and like going back. I've made an error somewhere. But I mean, this is a perfect teaching opportunity of like, this is what can happen essentially. And this is why you have to like quintuple check everything before you send it in. Because if I'd sent this in, I would have got something that doesn't match my file, and that's no good. So yeah, that's right. So I'm gonna have to go back in, figure out what I did. I think what I did was I probably added some extra letters at the front. So the way that this particular backbone works, if you don't want to mess with it too much, is it basically comes with a chunk of protein already being expressed by default. So in order to make my protein, this one, the pink one, fit and function with the backbone, I essentially need to make sure that it's what's called in frame, and there's no stop codons between the bit that I'm pasting in and the bit that I wanted to actually make. So I must have changed this both to add a restriction site here, I don't know why, presumably to make it easier to cut it out, but also perhaps to make sure that this is in frame. So do you see this chain of letters at the bottom here? That's the actual amino acid sequence of the protein. So if this is wrong, like if there's for example, let me just show you what happens if you do this wrong. So I added an extra letter, and I'm going to undo that in a second. But you'll see how now the sequence is completely changed. There's all these little stars. The stars are stop codons. This is very, very bad. Making this sort of mistake would be very, very bad, because you'll get nothing. Like nothing will be made properly. So please undo. Okay, good. We're good. We're good. We're back to normal. So I think what I probably did was I added some extra letters to make sure that this all lines up properly. But I don't remember what I did, and so this is gonna have to this is gonna be, you know, make myself a nice cup of tea and stare at this for an hour and just figure out what I did wrong. So considering that, we're gonna move on to the next thing, and it'll probably be the last one for the night because I'm tired and I've been talking for 2 hours. But I do hope people have been enjoying this, and I am looking to get back into doing streams more regularly. So yeah, and I was thinking about doing streams on another channel, just because, you know, sponsors were getting confused. They're like, why are these videos got so many fewer views? I'm like, because they're live streams. That's just how that goes. You know, also I was worried it was messing with my numbers, but I've figured if I just stick a giant this is a live stream label on the thing, that should differentiate it enough, and I can just leave it on the main channel and, you know, just make playlists. And I might unlist I think what I might do with the streams is unlist them, but keep them, like keep a playlist of them, just so that it's less confusing. So that way, if you want to find the streams, you can go to the playlist and just look for the one that you're looking for. But that way it doesn't like clog up the rest of my channel page, because it makes it hard to find my actual videos. So, you know, just something to think about. Okay, like I said, we're going to do one more quickie, and then we're going to get to it. So this is the protein called tyrosinase. Now, why is tyrosinase interesting? Well, I happen to have this is E. coli that is expressing this exact protein. So what tyrosinase does is it takes tyrosine and it basically chains them all together into a growing lump of tyrosine. Now, this probably doesn't sound too familiar until I tell you that that's what melanin is. So melanin, of course, the dark pigment that gives skin color its shades. It's what makes hair black or brown or red. Although red is eumelanin, it's slightly different. Not getting into that. Point is, if you make bacteria or yeast or whatever express tyrosinase, and you give them media that is chock full of tyrosine, they turn this lovely black color or brown or whatever. I don't know why certain colonies get super duper dark. Haven't figured out why that is yet, but it is the way it is. So this particular E. coli was designed by a friend of mine, and I asked him for the sequence that he used, and he sent me this. Now, the reason I asked for the amino sequence, not the DNA sequence, is because I want to make literally exactly the same thing, but in yeast, because I'm a yeast guy. I like me some yeastes. So I'm going to copy this backbone to melanin yeast. There it is. Copy. Lovely. Now we're going to go over to melanin yeast, which should be here. Cool. We now have the two things that we're going to need. Oh, okay. Thank you, Ember Beast, for the suggestion. Okay, so there's a question as why is this useful? One of the reasons that this could be useful is you could make a black beer. The other reason this is useful, like I just want it in yeast because I hate E. coli. They're so gross. I think it's a really interesting thing because you can basically show that it works at certain temperatures, but it doesn't work at other temperatures. I also just want to see how much melanin you can make, because melanin is one of the few natural black dyes. So, you know, if you could make enough of it, you could use it as a pigment, you could use it as all kinds of stuff, and yeast is a great way of doing that. I also want to, the reason I'm making this, I think it'll be a great thing in a kit. It's a great learning tool. It's something other than a chromoprotein. Like black is sort of weird. It means you got to make special media. So, you know, you're getting into like custom media that has different stuff. Well, thank you, Crackpot. So, you know, I think it's just interesting, really. I also want to compare it. The reason that my friend was working with this in the first place, and the reason that I was even thinking about this, is he's making rainbow flowers by expressing different chromoproteins in them. But he wants to make it CMYK. And so for that shade value, he's going to use melanin. And so he's making this, which is melanin production, presumably in the flowers, to make the first true black flowers. I don't know if this is any use in yeast, but I think it's neat, and so I'm hoping to have it made. I don't know if I'm going to have it made right away. I mean, you know, DNA synthesis, as you saw, is very expensive. But it's something that I think would be a great addition to the store, is just like a, you know, fun addition. And if you want this thing ready to go, I think look for a tiny green cell on Etsy, or it might be Bionomica Labs, one of the two. It'll come up. He's already selling this in E. coli if you want it, so have at it. Cool. So To turn this into a useful plasmid, first we need this in DNA form because this is useless. So what we got to do is we want to highlight all of it and click back translate. So basically what this is going to do, the program is going to, oh, thank you, Megalomaniac. Greatly appreciate it. So yeah, basically what this is going to do is it's going to take this amino acid sequence and translate it back into DNA in the species that I tell it. So I just scroll down, I pick Pischia, I can ignore all of this crap. I want to avoid EcoRI. Come on, fucker, there you are. And I want to avoid Naughty, because those are the ones I'm going to use. Do I want to make this with a his tag? I might put a his tag on this, just for fun. That could be fun. What do I have currently that has a his tag on it that I can show on stream? I don't know, I'll sort it out. Anyway, so preview optimization. And now this is gonna think for a second, load good, GC content, Uridian content, great, lovely. It's gonna save it in the right folder, great, cool. So, oh, there's an AVR2 there. Balls, okay. First things first, now that we have our bit of DNA, we go select all. First, we got to annotate, because fucking annotate your code. There's nothing worse than not annotating your code. Tyrosinase, thank you very much. Make it this lovely chocolate brown. Save annotation. Cool. Now I want to go make a translation. Cool. Now I need to add a cut site to both ends. So I already have this one because I use it all the time. We need this, copy, whoopie-doopie-doo. And I know that it's safe to copy this in directly because of the plasmid I'm using and where it cuts. So if I go translation forward, good, perfect. So all that mumbling is basically to say, I know that if I use this restriction site where I'm putting it, it'll all be in frame, it'll all work great, it's not gonna be a problem. So now I can scroll down, scrolly, grab this one. Again, it's one I use all the time, copy. Now, there's one problem with this gene as it stands. If I were to just do this, got my restriction site, the gene doesn't know where to end. So it also needs an ending. So I always do the same ending, which is TAA, TAG, TGA, and that's usually fine. Click enter. Now if I go translate, create translation, forward, yeah, it's all stars. So this is all stop codons. This is basically guaranteeing that the protein ends here. Like it won't keep going by accident, because that would be very bad. Now I need a his tag. His tags, his tag sequence. It's 6 of them. Okay, cool. So I need 6 histidines, which are these. So let's go codon tape up. You know what? I'm going to just copy them out of here. So let's just grab this, copy 1, pasty paste. Then I got 2 here, so I'm going to copy. My goal here is to get 6. What the hell? What happened? That's not good. Something broke. Something is very, very broke. Create translation forward. Huh. Don't know what happened there. That's supposed to be his. What the hell? Okay, this is... Oh, I see what happened. Okay. Got an extra letter. Don't know how that happened. Huzzah! Okay, lovely. That was weird. Why do I have all this extra crap on the end here? What is this? Oh, that's supposed to... Okay, I see what happened. I made a boo-boo. There we go. I knew something... I misclicked and I put the thing one spaced. This is what I'm talking about with like frame shifts and stuff. If you're one letter off, it breaks the whole thing. It's really annoying. Okay, let's try this again. Now we're going to copy this. We're going to go here. We're going to paste it in. So now I should have 6 histidines all on the end. Now, basically what this does, so histidine is sticky, for lack of a better term, basically. They're sticky. But they only stick to nickel. So basically, you can get, I mentioned this earlier in the stream, you can get a column that's full of these special nickel complex bead things. And when you run your solution with all the proteins in it through that column under a certain pH buffer, whatever the fuck, the his tag will stick to the nickel and everything else will drain through. So basically you'll end up with a column that has just your protein of interest stuck to it, and then you can run another solution through it that makes it let go, and then you end up with a solution of pure protein. Now, the reason that I'm doing this, I don't know if it's a good idea to put the his tag on the front or the back. Unsure. We're going with one. We'll work with it. There's a restriction site here, so if we ever needed to, we could cut there, or cut it out, rework it. It's annoying, but it's workable. Anyway, so the reason that I'm doing the his tag is so that if I wanted, I could grow the yeast, produce this protein, and lots of it, isolate it using the his tag, and then take the purified protein and just drop it into a solution full of tyrosine, and it'll just make melanin. Like shitloads of it. Instead of having to grow it with the organism, because then you're constantly worried about, you know, the organism producing enough of it, or, you know, dying because it doesn't like when you fill it full of tyrosine or whatever else. So it's just it's an added bonus for maybe something we'll use later, maybe we won't, we'll see. Hopefully it doesn't break it, we'll find out. Cool. Now we've got all of the features that I want in this thing. So now I can go, I can select naughty, I scroll up and I select eCoRI, and I go copy. Now this is my backbone. I'm going to rename it before I do anything, which is going to be Tyrosinase PPIC 9K. Always label everything. Cool. Now we go to the multiple cloning site. We collect eCoRI, naughty, pasty paste. And now you shall see, I can get rid of this translation. No, I don't want to delete the bases. I just want to delete the translation. Where'd it go? There it is. Cool. So now you can see that when I pasted it in, the sequence maintained exactly how it's supposed to. It's perfect continuity, which means that from the start of the protein here all the way to the end, it will be produced properly. This is very good. And because of the backbone that I'm using, I just happen to like this one. It's going to secrete this enzyme. So basically the media around the yeast is going to fill with this, because one of the issues was that tyrosine is hilariously insoluble. Like hilariously insoluble. It's tyrosine soluble. Let me just elucidate how insoluble this shit is. So in regular water, it's soluble at 0.45 milligrams per milliliter. So in a liter, you can dissolve at most 4 grams, which might seem like a lot, but that's nothing. So when I make, I can actually show you, I think. This plate is not white because of titanium dioxide, which is normally what I add to plates to make them white. This is actually tyrosine in the media. There is so much tyrosine in this media that it makes the stuff look white because it's not dissolved. It's just there. So one of the problems that I noticed with this stuff is that unless the tyrosine can get into the bacteria, it can't work. So what I figure is if I could just have the tyrosinase exude out into the media around it, it should work better, right? Like it can work on tyrosine that's just dissolved. It'll allow more tyrosine to dissolve more quickly. Um, as a ribosome, I can relate to looking at nucleic acid all day long. Funny. Thank you for the donation. Greatly appreciate it. But yeah, that's basically it. Done. Easy as that. Like I like it when plasmids are nice and easy like this. You just do the thing, a little paste, and it just works. It's great. I love that. As opposed to fucking this nonsense of this shit took me literally a whole day to like get to work. But this, it's nice, it's clean, clear, under control. Okay, let's delete that annotation. It is no longer helpful. Cool, so now we've got tyrosinase. So now, in theory, if I order this, I should be able to put it into yeast, and the yeast should just crank out melanin. Don't know why that's useful, but it might be. The one nice thing with this is I should be able to cut this out at any point. Like once I know that this works, I should be able to cut out the whole thing, stick it into a backbone for a food safe yeast, and then make like a black beer. Like a naturally black beer. I don't know if it's still going to be food safe because we've noticed that tyrosine. Okay, so when I grew, where the hell? When I grew this, it smelled funny. Not to say that, you know, a plate of E. coli doesn't already smell like butthole, but because it does. But this smelled real weird even for E. coli plates. Like it smelt weird. But when you look at tyrosine, just as a thing, you'll notice that this is what tyrosine looks like. When tyrosine is broken down, it's cleaved right here, which means this is what's released, which for those of you who don't recognize that molecule is phenol. Phenol is not good for you. So this is the one problem with growing shitloads of melanin. We think that just as a byproduct of having ridiculous amounts of tyrosine, it might be producing loads and loads of phenol by accident, which isn't great. Again, phenol is not good for you. So not sure what to do about that. Oh, Mr. Guacamole, thank you very much. I'm glad you enjoy. I'm glad I can inspire. Thank you very much. So yeah, I don't know if this is going to be food safe, but in theory it would be food safe. Just extremely high levels of phenol as a byproduct, not necessarily great. So yeah, but yeah, we're done. We're done. Huzzah! Okay, so as is tradition, I'm going to take a couple of minutes to answer some questions. So you made shake and bake phenol, yeah, kind of. So yeah, I'm gonna take a little bit of time. I'm gonna answer some questions. That's all the DNA designing we're gonna do for the day because I'm tuckered out and I need food. Starving. So let me just do the thingy. Now you get to stare at my beautiful face while I answer questions. And more to the point, I can actually see the questions in front of me instead of having to turn. Cool. So you got 5 or 10 minutes. If you got questions, now is the time, and then we're gonna wrap this up and call it for the day. What you got? What you got for me? Yeah, so phenol is kind of toxic, which is why it's not great for food. It's no good. There was a drug a long time ago, which they stopped selling because it made people heat to death, that was based on phenol. It was a phenol derivative that basically made mitochondria not work properly. But that was the point of the drug. Like I think it might have been p-nitrophenol, I'm not sure. But basically what would happen is your mitochondria wouldn't work properly, and so they had to go through a different process to make energy. So essentially you burned huge amounts of energy as heat. So it was a weight loss pill that essentially made you heat your fat away because you're just not, your nutrients aren't working properly. Yeah, that was super toxic for a variety of reasons, and one of them was people would heat to death. Like the therapeutic dose was very delicate. So if you went too high, you basically cooked to death. If you went too low, it didn't really do anything. So, you know, not ideal. Anyway. Okay, question time. What do we got? What got you into chemistry in the first place? Same thing that gets everybody into chemistry in the first place. It's like fireworks and stuff. It was just interesting to like see different chemicals reacting and doing stuff. I used to like playing with like copper complexes because they were real like transition metal chemistry is so pretty. But yeah. Okay, yeah, it was 2,4-dinitrophenol. Thank you. I mostly remember these things. Mostly. Okay. Top ways to get started at home. Fungus. Growing fungus. Like growing like oyster mushrooms or maitake or namiko or like lion's mane are fantastic ways of getting started with biology at home. By the time you've got all the equipment to do that, you're already like well on your way to doing other stuff, or like making wine or making beer. Like all of these things get you used to working with microbes. If you can do all of that, you're like well on your way, and then it's just a matter of building up your stock of genetic engineering tools, which not for nothing is expensive and time consuming, but like you can do it slowly over a lot of time. Okay, what else we got? Yeah. 2,4-dinitrophenol was not safe. This is why they don't sell it anymore. I think you can still get it online, and like some nutcases will take it as like a weight loss thing, but it's real dumb. What are your thoughts on computational biology? Do you have any interest in it? No. Those are my thoughts on computational biology. I don't know. It's not my thing. It's neat. There's been some really interesting stuff, but I'm a test it and see if it works kind of guy. Like I don't trust the Sims. All right, what else we got? What do you think about making the reflectin protein? Was that the octopus one? I feel like that's the octopus one that makes them shiny. I had forgotten about that until right now. I'll think about it. I've been thinking about how to do night vision biologically, and that could be an interesting way to do that because we don't have a tapetum lucidum. Like we don't have the shiny bit in our eye that like amplifies light, so we're shit at night vision. So I've been thinking like, how could you do that? I don't know. Reflectant could be a good way to do that. Homogenizer, fermenter. A fermenter, you can literally look for a glass carboy on Amazon. They sell them in every size. For a homogenizer, immersion blender. Works great if you've got enough stuff. It'd be gross if you're doing a collab, but I mean it would work. What else we got? If you want to learn DNA and plasmid design, what are the videos we should watch from your growing library? The Make Bacteria Glow one, the Make Yeast Produce Beta Carotene one, the Spider Silk one, maybe the PCR one would be good. I think I have a playlist. I think I have a bio playlist. Just watch the whole thing. Bio is broad. The secret to being a good biologist and doing what I do is learn as much shit as you can cram into your face hole. Like, literally, like textbooks on textbooks on textbooks of information, but like knowing which of that information you need at any one point is almost impossible. So, you know, start with like the manga guide to molecular biology, the manga guide to biochemistry, like start with those basics. Watch like all of Crash Course Bio. You don't need to memorize all of this stuff. You just need to have it available to you and just like listen to it once so that way you can draw from it from your memory if you need it, or at the very least you'll know what things to look up. Do you have a 3D printer? Oh yeah. Oh, I have so many now. I have 4. I've got the Prusa Mark III S. I've got the SL1, also from Prusa. I've got a Creality resin, and I've got the Phenom L. It's awesome. We've been printing massive things, and we're going to use it for kits and stuff later, but like at the moment we're just printing dumb shit. It's awesome. Okay, what else we got? What are you working on? Yes. Lots of things. I don't know. When do you think the lab will be ready? Probably the end of May. I'm going to start filming beginning of May. I'll have the first video out probably second week of May-ish, maybe first week of May, depending on like how like on top of shit I am. I'll have the second video out by the end of May, ideally, and then I'll do a lab tour in June. What else we got? Is Keg or Metapsych better for reviewing pathways? Do you know any better than these? I don't even know what those are. So, don't know. What else we got? I'm not answering that. Best way to contact me is you send me an email. Or if you want, become a patron and join the patron Discord. I answer that. Who is we? Oh, that's the other. So the new lab comes with new people. So I've got a small team of people that I'm working with, and you'll probably meet some of them. Some of them are a little camera shy, so you may not. But there is a collective of people now. So specifically, most of them are focused more on like helping to get the store set up, but some of them are going to be helping with videos. Some of them are going to be helping with the Silk thing. There's a lot going on. But so there is we, not just the royal we, but like, now we've got a team, which is, the other really exciting part of the new lab. And I'm very, very excited to just make all of the crazy stuff, because my team are the best. Like, they're literally the smartest people I know. And I was like, let's get all the smartest people I know in a room and let's just do really cool shit. And we are doing that, and I'm so excited. What fun mushroom cultures do you have lying around and I could get off you? The get off you bit, not yet. Although, okay, so one of the other things that's going to be in the store is base organisms and also just interesting organisms that I think people might want. So this is going to be like P. stypticus, which is my favorite fungus. I keep a jar of the stuff next to my bed. It's the glow-in-the-dark fungus. I love it. It's glorious. It's so pretty. So we're going to be selling that. We're going to be selling R. palustris, which is a purple bacteria, all the base yeast strains that you're going to need to do any of the kits, some kinds of E. coli, all the non-patented ones, just a selection of stuff. So, in terms of what's the most fun mushroom culture I have, that one, and you'll be able to buy it from me eventually. Yeah, can't blame the ones that are camera shy. Yeah, no, I mean, they're whatever. They don't need to be on camera. There's no reason. You might like see them in the background at some point, but like they're not there to be on camera necessarily. Like there's one of them, maybe two of them, maybe, but yeah, beyond that, they got their shit to do. They don't need to be on camera. So what else we got? Are you Tim Blythe's brother? No, I don't even know who that is. So, yes, I keep a jar of glow-in-the-dark fungus next to my bed, and it's awesome. It's sealed, like they can't get out. They're not making spores or anything. It's just a jar of I think I posted about it. One second, let me just. I have a picture of this somewhere. Where the hell? I haven't posted about it in a while. Oh yeah, here we go. This is it. It looks even more glowy now, and like you can see it with your bare eyes very, very easily. So the really bright bits are where it was trying to make a mushroom, and the mushrooms are way brighter. It just doesn't have enough room because I'm not opening the jar, so it can't make mushrooms. So they just kind of fizzle out. But while they're doing this like weird half growth thing, they glow very brightly. And so if I keep the jar next to my bed, it's all sealed, it's all nice. But when I'm like rolling around trying to fall asleep, I like turn over, glowing mushrooms. It's great. Okay, back to my beautiful face. They can't get out. You're making it sound like they can run. Clearly you've never worked with slime mold. Fucking thing can think and escape. Terrifying. I love slime mold, but it's so weird. Like, so weird. Somebody's... Okay. What else we got? Can you sell starter cultures for the glowing mushrooms? Yes, that's literally what we're going to be doing. Do you still have plastic-eating fungi? No, and no will not be available. It's really toxic. It's not supposed to be. No, no, hard no. Are you ever going to do more projects of getting pictures from satellites? Maybe. Before they encrypted it, I was thinking of doing one on getting the video feed off of the SpaceX launches, but then they encrypted it, so now it's illegal. So can't do that because they can't have fun. What price do you plan on selling it for? Not expensive. What half the thinking behind the store is that most biosupply companies are assholes. Like, there's no reason that, like, from ATCC to buy one strain of fungus is $400. That's insane. It's fucking yeast. There's no reason that it should be like this. So we're thinking like very reasonable, like $20, $25 maybe for a base strain. For a kit, there'll be like different tiers, right? Like depending on how much stuff you want. Like if you just want the DNA, it'll be like one price. If you want, the DNA, the yeast, the whole, like all the enzymes, everything, it'll be more. If you want all the media and everything, like, so there'll be different levels. If you want, we're thinking of, I think the deluxe kit with like all the media, everything is in the range of like 200, 250, but that's with everything. That's like all the media. It's literally the entire experiment ready to go, and it's a lot of stuff. Whereas if you just want the DNA, it'll be like $25. Like, go for it, have fun. Okay, what will the 3D printers be used for? All kinds of stuff. For the really big resin printer, which is the Phenom L, it'll be used for like producing like big parts for stuff. But we're also thinking of doing, making like props from like various types of media, like video games, movies, TV, whatever. Making a big prop, do all the 3D design, print it once on the full scale on the resin printer, then make molds of it and then sell like casts. So like if you want, like, so here I can actually show you. This was a test print that we did. This guy. So this disgusting little critter is the antlion grub from Half-Life Alyx. So we want to make a full-sized version of this and then like cast it out of silicone, basically, and then like sell the like wibbly wobbly grubs. Or we wanted to make it in the canister and the whole thing. So it'll be used for all kinds of stuff. Some of it will be just kind of jokey prop stuff. Some will be like useful tools. There's all kinds of stuff, and whatever we can think of, frankly. Yeah, Carolina is great for live animals. They're a little picky with who they sell to, so you just got to be, cognizant of that. That's really the only. So I mean, in terms of other fungi I'll be selling, it's mostly yeast. For those of you that don't know, yeast is a fungus. But yeah, so just various kinds of yeast, and the glowy one are the only fungus I'm really interested in selling. For like gourmet mushrooms and stuff like that, find a local place, or there's a million sites online that you can buy them from. I don't want to be that guy. Like I don't want to just be producing shitloads of mushroom cultures. The only reason I want to sell the glowy one is because it took me like 2 years to find somebody who actually sold a sample that actually glows. So I want to sell like the good shit. But yeah, anyway. Okay, I'm going to do like 2 more questions, and then we're wrapping it up for the day because I'm starving and I need food. What else we got? Okay, any way to buy lab stuff from companies that only ship to companies other than registering an LLC? No. Also, if registering an LLC is expensive for you, you're not going to have fun doing biology. Biology is so expensive. Registering an LLC should be like $100, maybe $200, which, I mean, it's not nothing, but like one reagent off of Thermo Fisher is like $1,000. One. Singular. So like doing the neuron project, pain, just pain, so expensive. So, you know, it's, I don't really have a better solution for you, unfortunately. We're hoping that eventually we're going to be able to sell. So one thing that we were thinking about selling, we're not sure if we're going to do this, is lab equipment with useless additions. So like think of a centrifuge, but it's full of RGB lights and maybe makes a noise. like you know, like as it spins up, it makes like a Star Trek noise or something, and like, you know, it does some lights. Just stupid shit. You know, we're thinking of maybe selling stuff like that, maybe, or actually like useful tools for a lab. But in terms of that, like it would be lovely if we could grow the store to the point where we're basically like a discount Thermo Fisher, but I don't see that happening. So we're going to start slow, start with kits, start with little stuff that we know we'll be able to get working, and then build from there. Okay, last one. Do you read papers on a screen or on paper? On a screen. It's just so wasteful to print out a thing just so I can read it. And I read so many papers in such a short period of time that it literally would not make sense to print them out. I would just have fucking stacks of paper everywhere. Because most of the time, like, I just want to burn through a paper really fast, like pull out the couple of pieces of information that I need and then move on. Like, I don't generally, like, unless it's something that I'm totally new to the field or I'm looking for something specific, I tend to just scan papers really fast and just, you know, move on to the next one, because I'll know very quickly if it contains the thing that I want. And when I do, it's mostly jump to the method section or look at the results really quick, scan them, see what I need, and then move on. I very rarely will read an entire paper, so printing the thing out makes no sense. But yeah. Okay, that was the last question. We're gonna call it there. Thank you everybody who donated. Thank you everybody who came and hung out. This was a lot of fun. I'm hoping to get back into doing more streams like more regularly. Yeah, we'll probably keep doing them on this channel. I was thinking of doing it on another channel. I think I'm gonna do it here. But yeah, we'll be doing more genetic engineering. I've got some interviews lined up with some really cool people who I think it's going to be really fun to chat with. And what I think I'm going to be doing though is I'll be unlisting the video, like the streams, but they'll still be up and available. They'll just be in a playlist where you can find them. So that way it doesn't, like I said earlier, it's not going to clog up my channel page. It's not going to make all the sponsors get all fucking weird about like your views are low. But yeah, so that's going to be that. Again, thank you everybody for watching. There's links to all the stuff down below. If you want to see how the lab build is going, follow me on Twitter, follow me on Instagram. I'm posting a lot on both of those places, so just follow me there. You can see how the lab is going. I suspect over the next week or two, I'm going to be posting a lot of photos of lab build because a lot of really cool stuff is happening over the next couple of days. So that's the best way to follow along and see how that's going. If you'd like to, you know, chat with me outside of streams, consider becoming a patron or a supporter. That way you can join my Discord and chat with me there. Other than that, I hope you guys have enjoyed, and I will see you on the next one. Have a lovely evening. Good night.